Hello and a very warm welcome to another lesson of science. How are you all? I hope you are enjoying my lessons, right? Let's go and see what's hidden in today's lesson. So students, we are going to revise the lesson plants, and the topic which we are going to revise under this lesson is photosynthesis and the experiments based on it. The learning objective for today's revision lesson is to be able to recall and remember the plants and the process of the photosynthesis by the special structure of the leaves. So if you see the outline structure of a leaf is something attached to the main stem of the plant with the help of the petiole. You can see this brown colored petiole, and this petiole extends inside the leaf in the form of a midrib, and from the midrib arises various branches which is called as veins, right? And the entire leaf is called as a leaf blade, and the margin, the tip, the edges are called as the leaf margin, and there's a tip of the leaf which is called as the leaf tip, and the entire leaf is green in color due to the presence of pigment called as chlorophyll. Now what we'll do, children, we'll take this leaf and we'll try to cut it with a pair of scissors, and then try to see the internal lateral structure of the leaf. How does it look like? So when we see the internal structure of leaf, the leaf has certain layers starting from the top, goes till the bottom, and there are different labeled parts of the internal structure of the leaf. The topmost part, which we can see, which we can feel, or the topmost surface of the leaf is called as the cuticle. Children, cuticle is a waxy layer. It's a layer made up of entire very thin layer of wax. What can be the reason for having a waxy layer on the upper surface of the leaf? The reason for this is to prevent the loss of water by sun's heat due to evaporation. Under this cuticle comes the upper epidermis. This entire blue color, thin layer, single cells, you can see single layer. This is called as the upper epidermis. Just under the upper epidermis, you can see the elongated or the long cells. The cells which are in length, which are in long shape, right? This is called, the word we use is elongated cells. They are green in color and they have dot green colored chloroplast in them. So these elongated or the long cells, they are called as palisade mesophyll cells, right? And the dots inside these are these dots. This dot, you can see here, children, this dot, these are the chloroplasts. As we come down, we'll see this light blue colored round cells which are not elongated like palisades, but they are round, almost round, we can say. They are called as the spongy mesophyll cells. You can see one of them here is labeled as spongy mesophyll cells. Okay, spongy mesophyll cells also, they have a chloroplast in them. You can see two chloroplasts are there, right? But spongy mesophyll cells, they are not on the top surface of the leaf. They are somewhere in the middle or down surface of the leaf. Since the top surface of the leaf or the upper surface of the leaf has palisade cells, and you can see in the diagram that the palisade cells has more chloroplast than compared to the spongy cells. Since the upper epidermis, under the upper epidermis, the palisade cells have more chloroplast. That's why if you observe the leaf, take a leaf, pluck out a leaf from the plant and try to observe the upper surface of the leaf and the lower surface of the leaf, you will find that the upper surface of the leaf is dark green, whereas the lower surface of the leaf is light green. So as we come to the last one, or the lower surface, that is called as the lower epidermis, children. Again, it's a single layer, okay, and it is called as the lower epidermis. Now this lower epidermis, it has openings at times, at places. So these openings are called as stomata. Now this is the opening, this is the hole, this is the pore from where the carbon dioxide enters inside the leaf, reacts with water inside the leaf, and forms glucose, C6H12O6 plus O2. Clear, children?
The amazing process of photosynthesis. A look around at nature is an absolute treat to our senses. We all enjoy the colors and fragrance of plants, but seem to forget about their important contribution. Plants are an absolute necessity for life on Earth. It is so because of the process of photosynthesis. A closer look at a plant shows that it is firmly held in the soil by the help of roots. These roots absorb water from the soil and through the tiny tubes transport it to the stem and then to the leaves. The stem holds the leaves in such a way that they absorb maximum energy from sunlight. A leaf is a very important part of the plant. Photosynthesis, the ultimate provider of food and oxygen, occurs in the leaf. A leaf consists of a flat, thin lamina, which is joined to the stem by a petiole. The flattened lamina provides large surface area for trapping sunlight. A cross-section of the leaf shows the upper and lower layers of cells, which are flat and irregular. Below the upper layer is the layer of tightly packed cells. These cells have specialized organelles called chloroplasts. Chloroplasts contain the pigment chlorophyll, which gives the green color to the leaf. The next layer has loosely arranged cells with large air spaces. The section also shows tubes specific for transporting the absorbed water into the leaves. The lower layer has many pores called stomata. Each stoma is surrounded by two guard cells, which regulate opening and closing of the stomata. A leaf absorbs carbon dioxide from the environment through these stomata, which, after passing through air spaces, enter into the chlorophyll-containing cells. Both water and carbon dioxide molecules are finally absorbed by the chloroplast. These chloroplasts also have the capacity for trapping energy from sunlight, and this energy is used to synthesize glucose and oxygen from carbon dioxide and water. These oxygen and glucose molecules diffuse out of the chloroplast. The glucose is transported through other specialized tubes to all parts of the plant, aiding in their growth. The oxygen present in these air spaces diffuses out through the stomata. So, during photosynthesis, oxygen is given as the life-sustaining gaseous component of the atmosphere, while glucose or food is transported to all parts of the plant and used for growth. To summarize, green plants can make their own food by the process of photosynthesis. Plants are held in the soil with the help of roots. These roots absorb water and send it to the leaves through specialized tubes. The lower layer has many pores called stomata. A leaf absorbs carbon dioxide from the environment through these stomata. Both water and carbon dioxide molecules are finally absorbed by the chloroplast. Where in the presence of chlorophyll and sunlight, they synthesize glucose and release oxygen. The oxygen diffuses out through the stomata. The glucose is transported through other specialized tubes to all parts of the plant, aiding in their growth.
So now we are going to do an experiment, and we will be taking up these tools and the apparatuses. We are going to take a beaker and a large beaker, and inside that beaker we'll fill some water, and we'll also put a green aquatic plant inside that water. That green plant is called as the pondweed. Now children, we'll keep this entire setup in front of the light. The distance between the plant and the light at first attempt will be 10 centimeters. Okay, that is from here to here. So my plant is here and it's 10. This is my first attempt. Okay, and I will record the number of bubbles given out. Now you all know children, bubbles are definitely, if some gases are given out, bubbles will be given out. So photosynthesis, if you remember, carbon dioxide plus water will give C6H12O6, that is glucose, plus oxygen bubble. So we will see when the beaker is at a distance of 10 centimeters from light, there was some amount of bubbles coming. Light is very important for photosynthesis, right? Now what if I increase the distance between the container and the between the distance of this container and the lamp? What will happen? Suppose I take the plant a little far away from the lamp, the intensity of the light will decrease, right? If the intensity of the light will decrease, do you think photosynthesis will happen faster? Photosynthesis will not happen faster. So what will happen? How do I know that the photosynthesis is not happening faster? I will see that the number of bubbles given out at this time will be lesser. Clear? So as we keep our bowl, as we keep the container away and away from light, the number of bubbles will also decrease. That shows that the rate of photosynthesis decreases if the distance between the plant and the light will be more and more. I hope you understand this, children. So after this experiment, this data was recorded there in the table. So first the distance was 10. So number of bubbles were recorded in 1 minute was 15. When the distance was 20, see, distance is increasing, children. So the number of bubbles recorded were 7 in 1 minute. When the distance is 30, more distance increase. So the number of bubbles are 3. Very good. In 40, the number of bubbles is 1, and in 50, the number of bubbles are 0. So you can see as we move the plants away from the water, from the light, the number of bubbles formed are becoming lesser and lesser, and these bubbles are of oxygen, which is the product of the process photosynthesis. So once you record your data in a tabular form, you're going to put this data in a graph. Remember children, I have discussed about the independent and the dependent variable. So we always keep the independent variable on the x-axis and we keep the dependent variable on the y-axis. What is the meaning of independent variable? Independent variable is the something which will not change because of some other variable. Okay, like for example distance. So distance between the between the plant and the light is not depending upon the number of bubbles. It's not that the bubbles are 5, so the distance will automatically change. Distance is stable. It's not depending upon anything, but the bubbles are depending upon the distance. If distance is less, more bubbles will be formed. If distance is more, less bubbles will be formed. So bubbles are the dependent variable. So as per our table, as per our data recorded in the table, we are going to put down the values here in the x-axis. So first the distance was 10 centimeters. Second distance was 20, then 40, and 30 is here. We have missed out that 30, then 60, and here in between 50, and then 70, 80, and 100. Okay, now the number of bubbles formed was, maximum bubbles was from 20, 15, 10, and 5. Now go back to the data, children, and just try to recall what you have recorded down in the data. So once more, this experiment is being explained over here. The same experiment, children, if you remember in your week 3 lesson or 2 lesson, I have shared the same worksheet with you all. I think it was there in the course pack also, right? So you can see here, Amna did an experiment for the rate of photosynthesis with respect to intensity of light. She changed the distance of the beaker from the light and counted the bubbles as per data below. You have to analyze the table and plot a graph. So this is, you can see, this is the plant kept right now at what distance? The plant is kept at 40 centimeter distance from light. So at first it was at 10, then it was at 20, then it was at 30, and later it reached to 40, and finally it was at 50, right? There must be some data recorded down. Yeah, we can see here. So as the distance is increasing, the amount of bubbles are decreasing. So let's see how to plot these things into the graph. First of all, let us identify what is the independent variable. Distance is the independent variable because distance is not going to depend upon the bubbles, right? But the number of bubbles, they depend upon the distance. If the distance between the plant and the light is less, the number of bubbles will be more because the light intensity will be more and the photosynthesis process will be also more. When the distance between the plant and the light increases, the number of bubbles will go down or decrease because the intensity of the light will decrease, and so the process of photosynthesis will also slow down or decrease. So once we are there with our values, with the data, we'll just try to plot these data on the graph on the x-axis and y-axis. First we have to label the x-axis and the y-axis, as we know that the independent variable come on the x-axis. So what is our independent variable? Our independent variable is the distance between the lamp and the beaker. So we're going to put this distance between the lamp and the beaker in centimeters on the x-axis, whereas the dependent variable, the number of bubbles released in 1 minute, which is depending upon the distance, it has to come on the y-axis, right children? And we'll just put down the numbers over there, just 10, 20, 30, 40, 50. We'll start from 0. Always start the number from 0 in the way while making the graph, and jump out 2 boxes, 10, and then 2 boxes, 20, 1, 2 boxes, 30, 1, 2 boxes, 40, and then 50, right? And then let us see the distance. We have our minimum distance is 0 and maximum goes to 15. So we can take this on this line. So it is this 2, 4, 6, 8, 10, 12, 14, 16, and our last number comes in 15 somewhere, right? So let us now plot the graph. I'm going to plot the graph in blue color. So for distance 10, children, for distance 10, what was the number of bubbles? For distance 10, the number of bubbles was 15. So my first plot will be around in the middle, right in the middle of 14 and 16. Is it clear, children? This is my first plot. Okay, somewhere here. Okay, what about the next number 20? When the distance was 20, the number of bubbles were 7. So this is 20 and my number of bubbles are here, right in the middle of 8. It's here. Yeah, it's here between 6 and 8. This is the 20. Okay, now what about the next one? 30. At 30, the number of bubbles were 3. So at 30, the number of bubbles are 3. So we have here, let me dark out this number also, 7, and let me dark it out 15 also. Okay. Now what happens? Let us see at 40, the number of bubbles are 1. At 40, the number of bubbles are 1. Okay, and at 50, the number of bubbles are 0, nothing. So now what happens? Now what we're going to do? Now we will join these plots, this dot with this dot, and this with this one, and then this one, and this is my final graph, which is coming out with the help of the data recorded in the table after the observation to the number of bubbles in 1 minute. I hope children you understood how to observe, how to do the experiment, how to analyze, how to plot down or record the data, and then finally represent your answer in the form of a graph by plotting and then later joining the dots and presenting the final graphical data. So finally, after plotting the graph, we are going to conclude our results. We are going to conclude our results based on the experiment, recording of the data and plotting the graph. We are going to conclude what we want to say when we did this experiment after our observations. So look at the question over here. Take the correct options to conclude the experiment. So we conclude that the rate of photosynthesis either increases or decreases with the increase between the distance of plant and light. With the increase between the distance of plant and light. So do you think the rate of photosynthesis increases, children? Rate means the speed of photosynthesis or the amount of the products made in the photosynthesis. So do you agree that the rate of photosynthesis will increase with the increase in the distance between the plants and the light? No, right? So the rate of photosynthesis decreases with the increase between the distance of the plant and light. Students, this lesson is very important. I hope you understood this lesson, and I hope you also understood how to do an experiment, how to record the data, how to plot the graph, and finally how to join to give a beautiful curve on the graph. I hope you're also clear now with the dependent and the independent variables, and you must be sure by now where to put while drawing a graph. I hope to see you all very soon, inshallah, children. Stay blessed, stay safe, and see you all soon. Bye bye.